we are fortunate. Oh, thank you. Okay, so today we are very fortunate to have Zabrina Brumme, who's going to be talking today, and I'll introduce her in a couple of minutes. But she'll be talking about immune responses to COVID-19 vaccination in people living with HIV. Next slide, Usman, please. So this is the last meeting before our holiday break. So we're going to have a number of weeks of vacation. So the week of December 27th and January 3rd meeting will be canceled, and then January 10th we'll have a talk from Melissa on long COVID, which is, as people know, a big focus of the group at this point, along with Recover Long COVID project team updates. So Melissa and Andrew, and then January 24th, associations between HIV infection and the clinical spectrum of COVID-19 based on our N3C data, and January 31st, findings, recommendations, best practice for multi-site EHR-based clinical research empowered by NLP, and that will be by Sun-Yang Fu and Hongfeng Liu. On the 24th, it'll be Jing Sun and Zhu Yang. Okay, next slide, please. So we're continuing with 69 sites having released data in the pipeline. 280 sites have DOA executed. That's really a lot. Next slide, please. With almost 3.5 COVID positive, 3.5 million COVID positive cases. And when I tell people about N3C, it's really remarkable. We're talking 3.5 million sort of 3-year windows of electronic health data. Next slide. So I think we're ready to introduce Zabrina. So very interesting background. She has a PhD from the University of British Columbia in experimental medicine, postdoc at institute that's between MGH, MIT, and Harvard, which was, I guess, at one point known as the Partners Research Institute. And she joined Simon Fraser in 2009 as assistant professor of molecular epidemiology. And so she has new investigator research funding, and her big thing is immune escape in HIV. So obviously the whole interplay between the immune system, HIV, and COVID is extremely timely and important. And so we're really looking forward to a really interesting talk. So Zabrina, I think this is the moment to share your slides, and looking forward to your talk. Thanks.
All right. Thank you very much for the very kind introduction and the opportunity to share some data today. I have just hit the slide share button, and hopefully you will see the actual slides and not the presenter view. Is that seeing it?
Yep.
Yeah. All right. Well, thank you very much. I'm not sure if disclosures are required, but in case I put them all here, our study has received some funding from the institutes showing and the agencies showing, and these are paid to my institutions, not directly to me. Other than that, I have no conflicts to disclose. A preliminary version of the data that I will present today is already available as a preprint shown here. We've recently further revised, added more data, and resubmitted the manuscript for consideration at a journal. But if you're interested in an updated manuscript, just let me know and I can send it to you. This was the rationale for undertaking our study. People living with HIV might be at risk of more severe COVID-19 outcomes. possibly as a result of immune suppression, but also possibly as a result of comorbidities or social determinants of health or other factors. Indeed, members of your group are among the groups worldwide that have shown this. So because of this, COVID-19 vaccination is important and expected to benefit this group. But at the time that we initiated this study, which was in April earlier this year, there was a number of knowledge gaps. First, there's some evidence from other infection models other than COVID-19 that HIV can blunt vaccine-induced immune responses. The number of people living with HIV enrolled in the Pfizer, Moderna, and AstraZeneca vaccine trials, which were the three major vaccines that were rolled out in Canada, those numbers were relatively limited. And at the time of, well, it wasn't until later this summer that the data was released for AstraZeneca, but at the time that we were initiating this study, there was no data available on immunogenicity outcomes in people living with HIV for Pfizer and Moderna. And again, at the time that we initiated the study, the real world data on the strength and durability of these vaccine responses in people living with HIV was also relatively limited. So our objective was to establish a prospective longitudinal observational study in the context of Canada's mass COVID-19 vaccine rollout, which lagged behind that of the United States and had some other differences. But our goal was to evaluate the strength and durability of COVID-19 vaccine immunity in people living with HIV. We enrolled 100 adults living with HIV through various clinics in the Vancouver area and also via community outreach. And all people living with HIV were receiving antiretroviral treatment. And in fact, essentially everyone had a sustained plasma viral load suppression. 95 out of the 100 individuals were fully suppressed at time of enrollment, and 5 people had a tiny little blip, but were otherwise suppressed. So it's a very, very well-controlled cohort. We also assembled a control group of 152 adults without HIV, and these ranged from 22 to 88 years old. I say we assembled a control group, not we recruited a control group, because about 3/4 of these controls were actually recruited for a parallel study that we were also undertaking on COVID-19 vaccine responses across the age spectrum. That cohort started to enroll as early as December 2020 and included healthcare workers, some folks recruited from the community. And we also recruited additional controls for this study. I'm mentioning this only because the timing of recruitment did influence what vaccines you got and how long you waited for your second dose. And I'll go into more details about that later. So this was the original plan for study visits and sampling. It's a longitudinal study where we tried as much as possible to have the first visit be before receipt of the vaccine. We were successful in the majority of cases. We collect serum, plasma, and we also cryopreserved PBMCs. We collect health and sociodemographic information by online survey questionnaires. And a benefit of this study that made it attractive for recruitment is we are returning the binding antibody results for each participant after each visit, so they can monitor their vaccine responses and also track any breakthrough infections that they might not have otherwise noticed. So this is the original study visit schedule. We had a pre-vaccine sample. We have a sample 1 month after the first dose, and again, 1 month after the second dose, 3 months after the second dose, 6 months after the second dose, and we were going to follow up again 12 months after the second dose. And one thing that I wanted to draw your attention to is here in Canada, because we had very limited vaccine supply for quite some time, Canada made the decision to increase the dose interval, the interval between dose 1 and dose 2, to 4 months or up to 4 months. But this variable kept changing depending on how much vaccine we had available in Canada. But some people waited up to 4 months for their second dose. All of the study participants received 2 vaccine doses between December 2020 and August 2021. Okay, this was the original study design, but of course, Boosters came along, and many participants are now eligible for third doses and boosters. Some of them have got them already. So now we're changing the plan, and we're following up 1 and 3 and 6 months after the third dose and the booster. Today, what I will show you is data from visits 1 through 4. That's a complete data set. And I'll also have a tiny little bit of data from visit 5, which is 6 months after dose 2, as well as 1 month after the boosters, because some people have received those already. The initial immune assessments that we have done have focused on humoral immunity only. We have collected PBMCs, but we just haven't had time or bandwidth to assess T-cell responses yet. That's coming, so today I'll only talk about antibodies. But in the data that I'll present today, there are 3 antibody measures that I'll be reporting on. First, just the total concentration of binding antibodies against the SARS-CoV-2 spike receptor binding domain, and we're using a commercial assay to measure that. This is the Roche Alexis anti-S assay. Secondly, I'll show data on the ability of these antibodies to disrupt the interaction between SRBD and ACE2. It's kind of a surrogate, higher throughput, sort of viral neutralization surrogate assay. And again, we're using a commercial assay to do this as the mesoscale diagnostics assay. And thirdly, we assess the ability of these antibodies to inhibit live SARS-CoV-2 viral infection of target cells in vitro. This is an in-house assay that we're conducting in the level 3 lab at Simon Fraser University. Our assays, namely the Roche Alexis assay, we're running both the anti-S as well as the original qualitative anti-N assay. So we're also able to identify participants who previously had COVID-19 prior to receiving their vaccinations. And we've also tracked we found a couple of breakthrough infections through this as well. So this is to remind me again, but I don't think I need to tell this audience, all COVID-19 vaccines encode the S antigen only. So vaccination will only produce anti-S antibodies. So if we find anyone with has both anti-N and anti-S, it means that they previously had and recovered from COVID-19. So I'm going to go through highlights of the participant characteristics in the 2 different groups. In the individuals living with HIV, everyone was receiving suppressive antiretroviral therapy. The median and the interquartile range of plasma viral load at the time of enrollment was undetectable. In fact, 95 out of the 100 individuals were undetectable. The other 5 were experiencing a tiny little blip, but I think the biggest one was 161 copies, and then they were previously and subsequently undetectable. It was also a very healthy cohort. The median most recent CD4 T cell count was 710 with an interquartile range of 525 to 935. The nadir CD4 counts were a little bit lower, a median of 280 with an interquartile range of 120 to 490. But we did have some individuals who had very low nadirs, nadirs of less than 10. For sociodemographics, the 2 groups were relatively matched in age, median of 54 for the people living with HIV versus 47 for the controls. That wasn't statistically significant. We did have differences in the sex distribution. There were far more males in the people living with HIV group compared to controls. We also had, sorry, the hospital system is going to be making an announcement. We had more participants who self-identified as white among the people living with HIV group than among the controls. The proportion of participants who had previously recovered from COVID-19 was the same in the 2 groups, about 10% or thereabouts. And we collected chronic health condition information by self-report, and the proportion of participants with at least one chronic health condition was 46% for people living with HIV and 33% for controls. We Chronic health conditions we defined as having one or more of 11 possible conditions. They're listed in red below. And the three most prevalent conditions were hypertension, asthma, and obesity in both groups. And here's some details about the vaccines. The vast 97% of controls received 2 mRNA vaccines. Most of those were Pfizer. That is because most of the controls were healthcare workers recruited at the beginning of the vaccine rollout. And at that point, mRNA vaccines were the only thing that were available in Canada. Most people living with HIV also received a dual mRNA regimen. But a substantial minority, 17% of people living with HIV received AstraZeneca, either for their first or both doses. And that again was because of the vaccine rollout. AstraZeneca began to be rolled out in about April as the mass rollout of vaccines was coming to Canada. And they were first reserved for older adults, but then rapidly it changed. And then after about a month and a half, AstraZeneca was basically no longer being used, no longer being recommended. So later on, the recommendation was anyone who had received AstraZeneca for a first dose should get an mRNA vaccine for the second dose. So that kept changing. And you can see that the two groups, the people living with HIV and the controls, are also different with respect to how long they waited between their first and second doses, with the controls waiting an average of 3 months and the people living with HIV waiting only an average of 2 months between doses. Again, this is because of the changing time between doses as the amount of vaccine available in Canada kept changing. The people who waited the longest in Canada for their second dose were actually healthcare workers who received their first doses pretty early, but then that was at the time where there wasn't a lot of vaccine available, so they waited the full 4 months. But those, for example, younger adults that got vaccinated later in the summer waited, sometimes 6 weeks only for their second dose. So we control for all of these factors in the multivariable analysis that I will present later. Okay, so here's the first data. Here I'm showing antibody concentrations after one vaccine dose. And here the cohort is split into 3 groups: people living with HIV who were previously naive to COVID-19 before getting vaccinated, control individuals who were naive to COVID-19 before getting vaccinated, and convalescent individuals, which are people who recovered from COVID-19 before getting vaccinated. And people living with HIV are black, controls are gray, and for the convalescents, they're in one group, but I've colored the dots so you can see what group each convalescent individual belongs to. And here, just looking univariately, there is a statistically significantly lower antibody response in terms of antibody concentration after one dose in people living with HIV compared to controls. And it's also it's about a 0.4 log without adjusting for anything, 0.4 log less. And as you can see, the level of antibody concentration after one vaccine dose in convalescents is very high, 2 logs higher than those of naive individuals, and that is consistent with many other studies showing a big boost even after one dose if you've had COVID-19 previously. Of course, there's many differences between the people living with HIV and control groups, so it's the multivariable analysis that's the really important one. So I'll show you the results of that. Here in the multivariable analyses, we controlled for HIV, age, sex, ethnicity. This was self-identifying as white versus not. The number of chronic conditions, whether or not you received AstraZeneca as your first vaccine. The next 2 variables are just relevant for the subsequent analyses, so are not relevant for analyses after one dose. We also looked at the days since the vaccine. This is because we were supposed to have done the sampling exactly 1 month after receiving the vaccine, but sometimes it was 28 days, sometimes it was 31, sometimes so on. So we just controlled for the number of days between the actual vaccination and the sampling, and whether or not you had previously had COVID-19. And here we can see that even after adjusting for all of these other factors, HIV infection still remains significantly associated with lower antibody concentrations after one dose, about 0.2 logs. But there's other factors that are more strongly associated with lower responses, and those are older age, so about a 0.1 log per decade decrease, and the number of chronic conditions. So each chronic condition was decreasing antibody concentrations by about 0.15 logs. You can also see here that if you're a convalescent individual, your antibody responses will be much, much higher. Okay, so this is what it looks like after one dose. But after 2 vaccine doses, here we can see on the left is the same graph that I showed previously, and in the middle now is 1 month after the second dose. And it's pretty clear to see that the people living with HIV group has almost completely caught up to the controls, still technically univariably significant. But pretty much almost everyone has a very robust response. It's almost even comparable to the convalescents. You can see that there are 2 individuals, one person living with HIV and one control participant who mounted very poor responses even after 2 doses. The person living with HIV is actually somebody who has an underlying blood disorder that leads to immunodeficiency. So there's immunodeficiency that doesn't have anything to do with their HIV. The control individual is a woman in her 80s with multiple chronic health conditions, including kidney disease. So those are likely to be the explanators for those poor responses. If we now do the multivariable analysis, here I've added in the middle 1 month after the second dose, and we can see that HIV infection is no longer associated with binding antibody response after 2 vaccine doses after adjusting for all of these factors. Instead, older age is associated with poorer responses, higher number of chronic conditions is associated with poorer responses. Having received 2 doses of AstraZeneca was associated with quite substantially lower responses, 0.6 of a log. There was also, our data also showed that longer dose interval, so waiting longer between the first and second doses, was also associated with a slightly higher antibody response as well. And that's consistent with data that came out of the UK. By the time 1 month after the second dose, it doesn't seem to matter if you're convalescent anymore. Everyone's kind of caught up to everyone else. Okay, so we had between the vaccine rollout and now, we've also had the opportunity to collect another sample 3 months after the second dose, and I'm showing that data here. Here we can see actually that the people living with HIV separate out a little bit more again. We're seeing about 0.2 or 3 logs less overall antibody responses in people living with HIV, and that's univariably significant. And the convalescents are staying a little bit higher. But again, we have to wait for the results of the multivariable analysis because there's differences between the groups. So I'll show those here. And here you can see that HIV infection is not significantly associated with responses 3 months after the second dose, but the p-value is 0.07. But what is associated with this sort of short-term durability, again, a higher number of chronic health conditions is associated with poorer responses at this short-term durability time point. Having received 2 doses of AstraZeneca continues to be associated with poorer responses at this short-term time point. And again, we're seeing that a longer dose interval is leading to maintenance of higher responses over time. Age is not statistically significant, but it's close. So this is just another view of exactly the same data that I just showed, linking everyone's longitudinal measurements. And it's just to sort of broadly illustrate that in general, you know, the trajectories in the people living with HIV and the controls are relatively comparable. It's also interesting that there's 2 individuals who, after the second dose, mounted a pretty poor response. And both of those individuals did eventually mount a little bit higher of a response. They were a bit delayed responders, but the magnitude of their response, continued to be the lowest in their respective groups. I'm happy to note that both of these individuals have subsequently gotten boosters, and their responses are significantly better now. We, of course, were interested in looking at the relationship between CD4 count, both the most recent CD4 count as well as the nadir CD4 count in individuals after living with HIV after receiving these vaccine doses. And here is the relationship between most recent CD4 count and antibody responses after one dose, that's in red, 1 month after receiving the second dose, that's in blue, and 3 months after receiving the second dose, that is in white. And here, nothing is statistically significant, but there is a weak positive relationship between antibody concentration and most recent CD4 count 1 month after the first dose, but really no relationship really between responses after having received 2 doses. It's important for me to point out, however, in our cohort, there are only 2 individuals with HIV who have a recent CD4 count under 200. So the CD4 counts were very high in this group. Here are the results for nadir CD4 count. Again, there is a weak positive correlation between antibody responses after 1 dose of vaccine and nadir CD4 count, that's the line in red, but there's no relationship whatsoever after 2 doses. So our interpretation is that for people living with HIV who are currently receiving therapy and whose CD4 T cell counts are in a healthy range, having had a low CD4 T cell count in the past will not necessarily compromise immune responses to COVID-19 vaccines presently. So I'll show the antibody function data a little bit quicker because it's in exactly the same format as before. So here are the data for ACE2 displacement activity after 1 vaccine dose, that is on the left, and 1 and 3 months after the second vaccine dose, that's in the middle and on the right. Here the axis goes from 0 to 100, where obviously the closer you are to 100, the better your function. And there's a grayed out area at the bottom from about 0 to 30. That's our estimate for what background in this assay is, and that's determined by testing plasma from individuals who are naive to COVID-19 and prior to vaccination, but really anything below 50 in this assay is pretty poor function. So here, 1 month after the first dose, people living with HIV who are naive to COVID-19 had significantly lower ACE displacement activity. So similar to the binding antibody results that we saw, convalescents were awesome after 1 dose. But after 2 doses, the gap between people living with HIV and the controls actually narrowed completely. In fact, 3 months after the second dose, people living with HIV appeared to have retained a slightly better activity, but that's, as you will see, that kind of normalizes out in the multivariable analysis. So univariately, it looks like after 1 dose, people living with HIV have a little bit poorer responses, but this goes away. Everything normalizes out by the second dose. And here are the results of the multivariable analysis. So 1 month after the first dose, HIV is significantly still associated with lower ACE2 displacement function. And there's a couple other variables that are associated with poor function, being male and having received 2 doses of AstraZeneca, same as the binding antibody results. And of course, having had recovered from COVID-19 in the past after 1 dose, that was associated with much, much, much higher antibody function for ACE displacement. One thing that I want to point out here that there's an additional variable that is in the multivariable analysis, and that is the anticoagulant used to collect the blood sample. That wasn't in the previous multivariable analysis because that analysis was done on serum and everyone's serum was collected exactly the same way. But because these analyses were done on plasma, and some were collected in EDTA and some in ACD, so we adjusted for that as well. After 2 doses, doesn't matter whether you look at 1 month or 3 months, HIV infection does not have any, is not associated with responses with respect to ACE2 competition, but some variables are. So older age is associated with poorer responses, both 1 and 3 months after. The number of chronic conditions is associated with lower peak responses 1 month after the 2nd dose. And again, having received AstraZeneca twice is associated with poorer responses. And again, here's the longitudinal view of those data. Just eyeballing it, it's relatively comparable trends between the people living with HIV and the controls. Here are the relationships between most recent as well as NADI CD4 counts and ACE2 displacement activity after 1 and 2 doses. It's quite similar to the results for binding antibodies. Nothing is statistically significant, but there is a weak positive relationship between this activity and both most recent and nadir CD4 cell count after the 1st dose, but there's no relationship whatsoever anymore after having received 2 doses. And the final function that we looked at was viral neutralization. This was a live virus neutralization assays where we're actually using not the Wuhan strain, but the Washington version of this is basically the Wuhan strain, but the isolate from Washington that happened early, very, very early in the pandemic. These are done in Vero E6 cells. This is not, we're not calculating an EC50. Rather, here the readout is the lowest reciprocal plasma dilution at which all 3 replicate wells are showing inhibition of cytopathic effect. So basically the higher the number on the y-axis, the better the neutralizing activity in plasma. And the lower limit of detection is also shown on the figures, as well as the upper limit of quantification. Basically, we go up to 2,560, and after that, we don't bother diluting anymore. So as you can see, 1 month after the 1st dose, pretty much if you're naive to COVID-19, most people will not show any neutralizing activity. There's a few who will be able to neutralize at 1 in 20, and some lucky few, maybe 1 in 40. But really there's not much going on there. So there's no difference between people living with HIV and controls who are naive to COVID-19. But we do see, as expected, that convalescent individuals, after 1 dose, most of them will have some pretty strong neutralizing activity. But as you can see, after the 2nd dose, whether you look 1 month after the 2nd dose or whether you look 3 months after the 2nd dose, there's no statistically significant difference between people living with HIV and controls. But the convalescents continue to remain higher, even after 2 doses. Here's the result of the multivariable analysis. After 1 dose, the only thing that's associated with neutralizing activity after 1 dose is if you've had COVID-19 before. That's pretty much because if you haven't had COVID-19 before, 1 dose isn't enough to give you neutralizing activity in most cases. But you can see that after 2 doses, HIV is not associated with the strength of neutralizing response. And again, it's kind of the usual suspects that are coming out. Age is associated with a significantly weaker peak neutralizing response, higher number of chronic conditions associated with lower response, peak response. Having received AstraZeneca twice is associated with a lower peak response as well as lower durability. And interestingly, if you are COVID-19 convalescent, you continue to maintain higher neutralization even 3 months after receiving your second dose than people who were COVID-19 naive. And again, this is the relationship between most recent and nadir CD4 T cell counts and viral neutralization activity. Here, again, after one dose, there is a correlation between better neutralizing activity and having had a higher, more recent CD4 T cell count, but it completely disappears. There's no relationships between recent and nadir CD4 count and neutralization responses anytime after having received 2 doses. Everything that I've shown so far was undertaken on the original Wuhan variant, which, well, is extinct. And these, at the time where we were preparing these data for publication, we were most concerned about the Delta variant. So we were able to assess ACE2 displacement activities and compare these between the wild type and the Delta variant. This was done using the mesoscale diagnostics technology. And here, 1 month after the first dose across the board, whether you're a person living with HIV who's naive to COVID, a control person who's naive to COVID, or somebody who's previously recovered from COVID, your antibodies are less able to displace the Delta compared to the wild type Wuhan variant, with all comparisons being statistically significant. But nevertheless, retention of some reasonable amount of ability to displace nonetheless. This is what it looks like 1 month after the second dose. Everyone is boosted up, but nevertheless, there's a statistically significantly lower ability to displace the Delta compared to the wild type SRBD, although there's still, at these peak response times, pretty good ability to displace the Delta SRBD. Short-term durability here, 3 months after the second dose, we can see that, as expected, immune responses and functions are declining as the amount of antibodies circulating goes down over time. But the ability to displace the Delta SRBD is still higher 3 months after the second dose, much higher than it was, say, 1 month after the first dose. But we concluded that vaccine elicited humoral responses might be less able to prevent infection by Delta in all people, regardless of HIV status and at all time points after immunization. And this is the last data slide that I wish to share today. This is very early data that we have on longer term follow-up, meaning 6 months after the second dose. And the very little data that we have so far collected 1 month after people have received boosters. As I mentioned, Canada was kind of pretty far behind the US in terms of the rollout, the timing of the rollout of the vaccines. The very first Canadians who were, acute care healthcare workers and individuals living in long-term care, those folks began to get vaccinated in December, just after Christmas, December 2020. But the Canadian population at large didn't begin to get their first doses until April 2020, and there it was age-based. So a young adult, people in their 20s, for example, didn't get their first doses until July. So we're just beginning. We haven't even collected the 6 month post-second dose on the vast majority of people living with HIV in our cohort, but because those folks all got their vaccines. They weren't prioritized for vaccines. People living with HIV got their vaccines at the same time. The rest of Canada was eligible based on whatever age you were in 2020. Nevertheless, we can see that in controls, as expected, the level of anti-RBD antibodies is going down over time between peak immune responses 1 month after the second dose and 6 months after the second dose. It's about a 0.7 log reduction in antibody concentration. But after a third dose, or the booster dose, antibody levels are boosted to levels that were even higher than what we saw 1 month after the second dose. This is also true in people living with HIV. So we can see that the people living with HIV, their booster doses, almost everyone is up at the upper limit of quantification. So higher than peak responses after 2 doses. Here, 6 months after the second dose in people living with HIV, we only have 4 individuals who have completed this time point already, and it might be tempting to say, oh, it looks like they're dropping faster, but there's only 4 dots, and 2 of these are individuals who mounted very poor responses to their first 2 doses. It just happened to be that we have data for them first. So I don't want to read too much into this very, very small amount of data. Take about a month for us to have a little bit more. And as with everyone else now, we're working on trying to assess the responses to the Omicron variant, but unfortunately, I don't have any data for you to show as of today on this particular cohort. So here's a summary of our main findings. 1 month after having received 2 COVID-19 vaccine doses, adults with HIV on suppressive therapy who had healthy CD4 T cell counts for the most part, they mounted broadly comparable antibody responses to people without HIV. Antibody responses waned after having received the second dose as expected, but after adjustment for age and health, then vaccine-related variables, there was no significant evidence that people living with HIV, their responses waned faster than those of controls, at least in the short term. We only have data out to 3 months. There was no evidence that a low nadir CD4 count negatively influenced the response to 2 vaccine doses, people living with HIV, but there was a number of factors that did. These were older age, a higher burden of chronic health conditions, and having received 2 doses of AstraZeneca. These were identified as negative modulators of antibody concentrations and function. Probably should have mentioned that the reason we did 2 doses of AstraZeneca versus not is because when we compare the responses of people who got 2 mRNA vaccine doses with a mixed regimen, meaning AstraZeneca followed by an mRNA vaccine, they're comparable, which is consistent with a lot of other data out there. It's really the folks who got 2 AstraZeneca doses that have really strikingly lower responses in terms of concentration and function. There's some caveats to our analysis. The specific thresholds that are needed for protection against infection and severe disease remain, well, we're starting to understand better, but we don't really know exactly precisely what those thresholds are. Our cohort was very healthy. Everyone was on stable therapy, and only 2 individuals in the cohort had CD4 counts less than 200. So our results may not be generalizable to people living with HIV who are not on stable therapy or who have very low CD4 counts. I only have antibody data so far. I don't have T cell data yet. And the durability data remains limited. We only have 3 months after the second dose at this point because Canada's vaccine response was pretty slow, although we're catching up now. The implications, people living with HIV whose viral loads are well controlled on therapy and whose CD4 T cell counts are in a healthy range currently. We interpret this to mean that these folks will not immediately require a 3rd COVID-19 vaccine dose, meaning a 3rd dose as part of their initial immunization series, immediately after the second. But other factors like age, comorbidities, type of initial vaccine regimen, longer-term durability of these responses, and of course the rise of variants, including Omicron, will influence when people living with HIV as well as people not living with HIV will benefit from booster shots. In fact, people are getting them now. And further studies of people living with HIV who are not on therapy and who have low CD4 counts are needed, as our study does not cover those folks. And there's a very large number of people who are on the study team. So I'd like to thank all of my colleagues, as well as our community representatives, our funders, and most of all, to our participants, without whom it would not be possible to do research. And thank you also for your attention.
Well, thank you. That's a really great talk. Very, very interesting. Questions from the group? So one thing that's your conclusions really seem to be that people who are well controlled by HIV therapy really can be, as far as you can see, and tell me if I'm wrong, viewed as not really compromised in terms of their response to vaccines, and really it's a testament to the efficacy of anti-HIV therapy.
That's correct. That's the interpretation that we made from our data. I should mention that the province of British Columbia, Canada, where we are based, is one of the few places where we've done a fantastic job of making sure that folks are engaged in care and on treatment. And so HIV incidence, in part because the treatment levels and suppression levels are so high, are steadily continuing to decrease year after year. So the population that we were able to study here are folks who are all on well-controlled therapy with high CD4 counts. But yes, the interpretation that we made is that if you are living with HIV, but you're doing really well on therapy, you can expect that your responses to these vaccines, these COVID-19 vaccines, is going to be strong, as strong as someone else your age with a similar health profile. That said, everyone's getting booster shots now, so we'll be able to. I don't know if we'll be able to really answer the question about long-term durability, because there was kind of this hint at 3 months that maybe the responses were strong, but perhaps not as durable. But I don't know if we'll be able to answer that question really in the long term, because booster shots have already started.
Interesting. So David Sonner says, thank you for the excellent presentation. Any interest in looking at the association between markers of immune activation and vaccine response in PLWH?
Absolutely. We want to look at cellular responses. We have cryopreserved PBMCs. We just haven't had the bandwidth to look at them yet because samples are still coming in. We're spending all the time processing them. And we're also interested to look at just cytokines and other things as well. So absolutely, that's in our plans.
Right. And then Julie McMurray says, how do the differences between PLWH versus control compare to flu vaccination, with the caveat that flu is single dose and not mRNA? That's an interesting question.
Yeah, we haven't had the opportunity to look at flu vaccination ourselves, but from what we understand from the literature, flu is one of those vaccines that often, I shouldn't say often, but there's some evidence in the prior literature that responses in people living with HIV could be weaker. That said, it's important to, there's a difference between people living with HIV who are not currently accessing therapy with folks who are very, very well controlled on long-term therapy. So I think it's important for studies to distinguish those two populations.
Right. No, I mean that would seem to be very important. And then presumably the level of control in some people can vary a lot. So it might look good at the moment, but there could be a history of poor control. And how does that impact things? Which I guess, since you're fortunate to have a population that's well controlled, you can't really look at that easily.
Yeah, everyone had been controlled for quite some time. There was one individual in our cohort who actually was recently diagnosed. We recruited this person in April, and they had recently been diagnosed with HIV, with likely having acquired the infection recently. So they had only been suppressed for about 6 weeks, only because they had only been diagnosed 6 weeks ago.
Right.
But everyone else had been suppressed for at least 6 months, if not longer. And we have many people in British Columbia who have been suppressed for, a decade or longer. So we have a very, very well controlled.
Yeah, that's impressive how effective that is. So question on Dr. Yang. Thanks for the informative presentation. I was wondering if you considered the impact of different types of vaccines on the immune response between PLWH and control groups.
Yeah, our study isn't explicitly designed to do that. That said, we are asking in our questionnaires about uptake of the flu vaccine and whether folks got it, and if so, when. So we might be able to do secondary analysis on that, but all of that information would be by self-report. As we know, sometimes people can misremember when they got a certain vaccine, or decide that the questionnaire is too long, skip some questions. But we might be able to look at that if we have sufficient enough responses to that question.
Great, yeah. And okay, question from Craig Wong. Any evidence of antiretroviral therapy affecting viral load of COVID-19?
Thank you for asking. No. We had a couple of anecdotal, a couple of physicians called us, oh, my client had a blip, and then we thought, oh, did your client recently become, did your client recently get vaccinated? And sometimes the answer is yes, and sometimes the answer is no. We did try to perform an analysis. We do have access to all the viral loads longitudinally for everyone in British Columbia. What we don't have is everyone's exact vaccination date and status. That is not something that we have access to, but we can, for example, just look at viral loads within a specific window of time and compare them to, another window of time prior to the advent of the vaccine. So we actually did this. We compared 2021 between April and August to 2019 between the same dates, thinking that if there was some large effect of the vaccines on viral load blipping, that we would see more blips, but we did not. In fact, it almost seemed like we saw slightly less blips. So it's not, our analysis wasn't designed to do that, but we didn't see any obvious evidence of viral load blips that coincided with the vaccine rollout in people living with HIV. But I'm aware that there's some reports, anecdotal reports, that this might be happening in some cases.
Well, wonderful. Well, I think we've gone through the questions, and this is really fascinating work and extremely well thought out, thorough, and incredibly timely. So thank you, and hope you and everybody have a great holiday.
Yeah. Thanks again. I appreciate it. Happy holidays.
Happy holiday. Bye-bye.